Hello, welcome to Aryamade. I am Arya. I'll teach you the maid. So today we have a very important topic which is considered to be acid-base balance. It's very important in order to understand. First of all, let's talk about what is acid and what is base. See, I know that there are lots of definitions of the acid and the base that we have to talk about, but we are not in chemistry. We are in biochemistry. So let's go with the least true. However, the easiest definition, acid is something that generate H plus and base is something that take H plus or generate OH minus. This is not much important, but this and this is very important whenever we talk about acid and base in our body. Okay. Now there are two types of acid. One is considered to be strong acid, another is known as weak acid. Strong acid means something that gets completely hydrolyzed. Like let's say HCl, it gets completely hydrolyzed to H plus and Cl minus. So in normal condition, back reaction is not possible. However, weak acid means they are partially hydrolyzed, like we have got H2CO3. That means it can get reversed if something happens. Like if the concentration in this side increases, the reaction shift towards this side. If the concentration here increases, the reaction shift towards this side. If the concentration here decreases, reaction shift towards this side. If the concentration here decreases, reaction would shift towards this side. So yes, there are a lot of dynamic condition. Similar goes for the bases as well. So weak acids and weak bases are the best for the buffer. Buffer means weak acid and weak bases that do not allow too much change in the pH. Okay, so we'll be talking about buffers as well right now. Right here today in this video. But first of all, let's understand why are we talking about acid and base balance in the body. What would happen if acid increases or base increases, or altogether what would happen if the pH changes? That means what is the significance of pH in the human body? One of the most important thing about the pH is that it causes tautomerism. How does it cause tautomerism? Because H is the one that moves during tautomerism, if you remember. Now, because of the tautomerism, what happens? There is difference in the H bond, and H bond is a very important bond whenever we talk about the nitrogen bases in the contents of our nucleic acid like DNA and RNA. So first of all, the nucleic acid is affected by pH. How? By affecting tautomerism. How? By affecting the H bond of our nitrogen bases, complement nitrogen bases. Next is proteins. Actually, we know that proteins are charged molecules. The structure of the protein as well as biological functions, they are dependent on the charge. And H plus is also a charged molecule. Molecule? No, atom, right? Now this H plus does influence the charge of our proteins, influencing the structures and the balances. That's the reason why enzymes, they have got their optimum pH. So yes, here comes the enzyme which affects our metabolic processes. Not only this, our lipids, our carbohydrates, our nucleic acid, all of their charges are affected by pH. And one more thing, very significant. Actually, this pH is even responsible for the oxygen transportation changes and carbon dioxide transportation changes. If you remember the Bohr effect, what happens? H plus increases, thus decreasing the affinity of oxygen, thus causing the oxygen unloading to the tissues. So this is also a very important feature of the pH. So that's the reason why pH is so much important, and that's the reason why we have to talk about pH. Now that means our body tries to maintain a proper pH. Actually, from 7.35 to 7.45 is fine. Normal body pH of the fluid is 7.4. I'm talking about the body fluid, especially the plasma, not anything else. In the cells, we have 7. In the RBC, we have 7.2. I said that 0.5 is tolerated, but the normal is the change still 0.04. Okay, 0.05 is tolerated, but best would be the change in 0.04. If you see the venous blood, in venous blood, the pH is about 0.36. And in arterial blood, the pH is about 37 actually. Okay, and in arterial blood, we have got 0.41. So the difference you'll be observing is 0.04, and it is normal for the body to function. What if the pH rises? Actually, you should remember that the concentration of H plus in our body is like 40 nano equivalent per liter. And similarly, HCO3 minus is in like 24 milli equivalent per liter. That means the amount of H plus is very low in comparison to that of the HCO3 minus. Right. So yes, this is also something we have to consider a lot. Now, let's come to the next part. Buffer. So I have already mentioned about the buffer. Buffer are our weak acid or bases that avoids large changes in the pH. However, they have some limitations, right? Because let's say we have a buffer, very important buffer, which is known as HPO3 2 minus, which when it gets H plus convert into H2PO3 minus. Now if it has like H plus, If the label of H plus is in a certain way, yes, the buffer does work. Or if H plus decreases in certain way, the buffer does work. But if it's in a high way, it cannot cope up because this molecule is also in a limited quantity, isn't it? Therefore, this is for the small changes in pH. If there is a high changes in pH, there will be changes in the body. That's why we are studying about acid-base balance, aren't we? Now, whenever we have to talk about the buffer, we divide the buffer into two parts. One is considered to be the chemical buffer and another is considered to be the physiological buffer. Now, let's talk about the chemical buffer. In chemical buffer, first of all, we have got proteins. In proteins, we have major two protein. One is considered to be the hemoglobin and another is considered to be the amino acid. So hemoglobin, it binds with our acid. Similarly, it also binds with our carbon dioxide, forming carb amino hemoglobin and hence acting as a buffer. Similarly, amino acid as well, it binds with the carbon dioxide or acid, forming the carb amino acid as well. Especially the amino acid that we talk about, let's say the protein that we talk about would be albumin because it is maximum in the serum. Exactly. Now another is our phosphate buffer, which I have already mentioned about. Now this phosphate buffer is actually important in order to take the H plus to the renal because it helps in the sodium and H plus exchange. And one of the thing is this is normally present even in the RBC, okay, or any cells. So what happens? So when H plus increases here, the H plus, I mean in the serum, when the H plus increases towards the blood or let's say intracellularly, H plus goes in. But at the same time, potassium comes out as well. So increasing the potassium in the serum. Similarly, if the H plus decreases, the H plus would come from the intracellularly to the extracellularly and potassium would have to go intracellularly causing hypokalemia. So we have to consider this pathway as well, this exchanger as well, which is considered to be sodium potassium exchanger. Now next buffer that we have got is carbonic acid buffer. Now this carbonic acid buffer is very famous buffer. Let's say you have got carbon dioxide in the presence of water. See, if there is presence of carbonic anhydrase or dehydrase, anything in any tissues, if they are present, then only this reaction occurs. In serum, this enzyme is not much. However, we cannot say it is not present. That doesn't mean this reaction does not occur. The reaction is very slow. That's why if carbon dioxide increases significantly, yes, the pH of the blood does decrease. But if it is in a little amount, the conversion is not much significant, and in order to make it significant, there is necessity of the carbonic anhydrase. So in the tissue where there is carbonic anhydrase or dehydratase, it's converted into H2CO3. Okay. Now this is a weak acid, so it dissociates into H plus and HCO3 minus. Now this is very important thing to talk about. Okay. If H plus increases, yes, the reaction will shift this direction. If H plus decreases, the reaction will shift towards this direction. So in this way, it acts as a buffer. Now that was about the buffer. Let's talk about the physiological buffer. Physiological buffer is very important buffer. But before we move towards the physiological buffer, I would like to mention about what are the sources of our acid and bases in the body, especially the acids. Because acids are very much present in our body. Like we eat food, don't you eat lemon? You get a lot of citric acid there. Any sour fruit, there is acid. So through the food as well, we eat a lot of ascorbic acid in our pickles as well, isn't it? And another is due to metabolic process. See, in metabolic processes, there is formation of lactic acid, acetoacetate and other keto acids as well. And similarly, acetic acid and so many things, propionic acid, butyric acid, they all are produced during our metabolism as well. So metabolism is also responsible for increasing the H plus concentration. Another is our carbon dioxide. As I have already mentioned, we breathe oxygen. As we breathe oxygen, our body would produce carbon dioxide, which also increases the acidity of the blood. Then next that we have got is sulfur-based amino acid. This sulfur-based amino acid, when they are metabolized, they form sulfuric acid. Now this sulfuric acid are also responsible for increasing the acidity of the blood, or let's say decreasing the pH of the body. Now see, I was talking about the amino acid. We have to remember in amino acid what happens. See, if there is a neutral amino acid, the amount of H plus formed will be equal to amount of H plus utilized, especially during the formation of urea. So neutral amino acid, not much important. Then there is dibasic amino acid. When this dibasic amino acid are metabolized, they produce H plus. However, dicarboxylic acid, when this dicarboxylic acid are metabolized, they decrease the H plus because we are metabolizing acid here. When the acid are metabolized, they form base most of the time. And sulfur containing amino acid like we have methionine and cysteine, they increase the H plus. So this is very important when it comes to the amino acid. Now let's talk about the glucose metabolism. See, in the glucose metabolism, during glycolysis, yes, there is a formation of H plus, isn't it? If there is lactic acid formation, there is even more formation of H plus. That's there. In fatty acid, if you are doing lipolysis, there is generation of H plus. If you are doing formation of ketones, there is generation of H plus. But during utilization of ketones, the H plus are utilized. During esterification of our fats, let's say esterification, in this case H plus would decrease. So in our metabolism as well, H plus is playing such an important role, and in metabolism as well, a lot of H plus are generated and utilized. So the source of our acid and base are actually internal as well, not just the external acid that we intake. Now finally let's move towards the physiological buffer. Actually physiological buffer work only when the chemical buffers do not work, and in physiological buffer we have two buffer. One is considered to be a respiratory buffer, another is considered to be a renal buffer. Actually respiratory buffer is very fast. It takes minutes to hours for the respiratory buffer to occur. However, renal takes hours to days. That means it takes a longer period of time. In respiratory buffers, we know that carbon dioxide is removed out. As the carbon dioxide is removed out by hyperventilation, our body actually balances out. Like we know that H plus is detected by mostly by the peripheral receptor. When the peripheral receptors they detect the H plus, even central receptor also can, if the H plus concentration is in a huge amount, there will be hyperventilation, and in that way our body can maintain. If H plus decreases, our body would note that as well, and because of that reason as well, there would be hyper. Once the CO2 is retained, the acidity would be back to normal. Now there is renal. Now in renal, what do? We even excrete acids which are non-volatile acid. Volatile acid like CO2 are removed by our respiration. So H plus like non-volatile acid, or HCO3 minus like bases can also be removed through kidney. Anything is possible. Now, when we talk about acidosis, acidosis means the condition in which the acidic environment of the body increases. That means H plus increases or pH decreases. We divide acidosis into two parts. One is considered to be respiratory acidosis, another is considered to be metabolic acidosis. So first of all, let's talk about respiratory acidosis. In respiratory acidosis, due to hyperventilation, our CO2 increases. This is the respiratory acidosis. In order to compensate it, our renal conditions like our renal or our kidney, they excrete the acid out, or they simply reabsorb the HCO3 minus. Now this is very important. What happens after HCO3 minus reabsorb? This is a little bit part of our respiratory physiology as well that I wanted to include here. Okay. Actually, as the HCO3 minus is taken, the blood HCO3 minus will increase. Now let's say this is the blood-brain barrier and this is our CSF. In the CSF, we know that carbon dioxide plus water give rise to H2CO3 minus, H2CO3, which further give rise to H plus and HCO3 minus. Now this H plus, because it's a huge amount in the blood, it starts traveling here. As this increases, this H plus in turn decreases because the reaction would turn in this way. So as this H plus decreases, it causes hypoventilation. So see the problem here. You got respiratory acidosis. What happened? Your HCO3 minus, you start absorbing a lot. It can even cause hyperventilation. This is considered to be the desensitization of our central chemoreceptors as well. And in this case, peripheral chemoreceptors are the major worker. Okay, so this is very important thing to remember. So in respiratory acidosis, renal has to do something. Renal has to remove out acids, or they have to simply absorb a lot of SCO3 minus. If it is compensated, you will find SCO3 minus increasing. If it is not compensated, you will find it normal. And in respiratory acidosis, you will find CO2 more and pH less. Okay, so this is very important. Now let's talk about metabolic acidosis. In metabolic acidosis, there can be two conditions. One is increase in the number of non-volatile acid, such as our lactic acid. And another would be decrease in the number of SCO3 minus, like there is a lot of excretion of SCO3 minus, or there is lack of absorption of SCO3 minus, especially in renal tubular acidosis 2. And one of the thing is like lack of production of ammonia and all those things in the context of acidosis. That would be our RTA1. Okay, so in these conditions, and when there is a lot of volatile acid, there is a high chance of metabolic acidosis. And in metabolic acidosis, what happens? Your body try to compensate it by causing hyperventilation, okay, because this H plus that is present a lot, this H plus would go to the peripheral receptor, and as they go to the peripheral receptor, they would cause the hyperventilation. Okay, so this is very important thing to talk about. Now let's talk about alkalosis. Now talking about this alkalosis means decrease in the acidity or decrease in H plus or increase in pH is considered to be alkalosis. It is also two types. One is considered to be respiratory alkalosis and another is considered to be the metabolic alkalosis. Now talking about this respiratory alkalosis, what happened? The carbon dioxide decreases. How? Due to hyperventilation, it decreases the CO2. Now in order to compensate, our body starts excreting out HCO3 minus. So this is very important. Okay, now let's see what happens in the central receptor. Because HCO3 minus starts being excreted out, or sometimes the H plus is reabsorbed, in that case altogether the concentration of HCO3 minus would decrease. So in the CSF we have got H2CO3, which is forming H plus and HCO3 minus. This starts coming here. As this starts coming here, it decreases and H plus increases. Now this causes further hyperventilation, and there is further hyperventilation occurring. So this is very important, further decreasing the CO2. Now this is a very huge problem here our body is facing. Okay, rather than compensating, our body is further creating a lot of problem. Now let's talk about it. So in this way, our renal would try to compensate it by removing the HCO3 minus. So in the respiratory alkalosis, you will see decrease in CO2, increase in pH, and if it's compensated, then decrease in SCO3 minus as well. Now let's talk about metabolic alkalosis. In metabolic alkalosis, what happens? The H plus is excreted out a lot, or HCO3 minus is observed a lot. In both of these cases, metabolic alkalosis occur. So in this case, you will observe that HCO3 minus has decreased, pH has increased, and if there is compensation, then in that case, CO2 would increase. And this is done by decrease in H plus, means it would go to the peripheral receptor, and this peripheral receptor would ask for the hypoventilation, and in this way our respiratory signal would actually cause hyperventilation and compensate for this metabolic alkalosis. Now one of the thing I would like to mention about it, that is anion gap. Okay, why are we talking about anion gap? Why is it so important? See, I said that in metabolic acidosis or alkalosis, both volatile acid as well as SCO3 minus play a role. So how are we supposed to know which is the one that is responsible? For that we have anion gap. What is anion gap? We take sodium and potassium from cation or any components that are cation in nature, okay, that are positive charge in nature, and we take SCO3 minus and Cl minus as anion. Now what do? We subtract cation minus anion, and this is considered to be anion gap, which should be plus minus 10 millimole per liter. Okay, so when the problem is with H plus, the anion gap would increase because we have this anion gap. In anion gap we'll have proteins and extra negative charged components, and if this H plus would increase acid, in that case anions would be there. Like if you have got lactic acid, there is lactate as well, and lactate would be an base, so that would increase anion gap. Okay, however, if the component is SCO3 minus, let's say SCO3 minus is excreted from the kidney, then in that case it would compensatorily absorb the chloride, not letting anion gap to change. At the end, I just want to give you a little bit summary. If it is acidosis and if there is alkylosis, okay, I'll give you a little bit of summary. The pH would always decrease, and in alkalosis pH would always increase. If it is respiratory, then in that case CO2 would increase. Here also, if respiratory, then CO2 would decrease. Okay, if it is metabolic, then in that case HCO3 minus would decrease and SCO3 minus would increase. If it is compensated, then SCO3 minus would increase, CO2 would decrease. If it is compensated, okay, that is just opposite that you would observe, and your SCO3 minus would decrease if it is compensated, and CO2 would increase if it is compensated. Be careful, it could be normal as well as compensated, and this is very important. Okay, so that was all from the acid and base balance. I hope you enjoyed. If you liked it, give a big thumbs up. If you did not, write down in the comment section what do you want me to improve. And yes, stay curious, stay learning. Bye bye.